Thank you very much, Lisa. Can everyone hear me okay? Great. It's a real pleasure to be able to come here and speak to such a great audience tonight. What I wanted to do in this, over the course of the next 45 minutes or so, is go through some of the work that my lab has been doing in collaboration with a large number of investigators around the world to pull together very large collections of genetic data from humans, and then also think about the ways in which we can apply those massive data sets to understand rare diseases. As Lisa mentioned, that's really the big focus of the lab. And I'll broadly divide the talk up into three sections. I'm going to spend a fair bit of time at the beginning here. I know the background of the audience here is very mixed, so I'm going to go over some fairly basic stuff that gives you some information about the type of data that we can actually extract from a genome sequence, and really encourage you, I think, to think about the genome as a way of storing information, storing and displaying a type of digital information. The second section of the talk, relatively briefly, I'll go through the challenges and the opportunities that are posed by the diagnosis of rare diseases. So these diseases that are individually extremely rare, often very severe, but collectively affect somewhere between 5 and 10% of the US population. And then the final section of my presentation, which I guess is really the focus of the work that we do here, is really emphasizing how we can actually use very large collections of genetic data, not just from rare disease patients, but also from the general population to improve our diagnosis of patients and families affected by these diseases. So let me start then by talking about the type of information we can actually extract from a DNA sequence. Now, I'm not going to go through exactly what DNA is. I think all of us have been steeped in a sort of intuitive folk knowledge about what DNA is. We know that it's a physical molecule, but also that it carries some type of information. And of course, this molecule is shown in this iconic representation here of a double helix. That's actually what it is. It is actually a double helical molecule that's present in every cell in your body. And this double helical molecule contains these rungs or strands that correspond to individual letters of DNA. And those are one of only 4 different types of letters that are present in your DNA, A, C, G, or T. And that A, C, G, T repeated a mind-bogglingly large number of times in many different arrangements is what provides the information that allows our cells to function. So this is an actual snippet of human DNA. I'm just going to let this flash past for a second here. Now, at peak speed, that little piece of DNA was going at around about 50 letters per second. And so it's worth pausing a little moment to understand just how staggeringly large the genome is. Despite the fact that it's condensed into every single cell in your body, it would actually take, if we were to play it at that peak speed of 50 letters per second, it would take us about 2 years to run through one strand of your genome. If we wanted to read both pieces of your genome, that is the section that you've inherited from your mother, the section you've inherited from your father, that's 4 years playing that particular movie. So this is an enormous amount of information that is recorded again in every single cell in your body. That's 2 by 3 billion letters of DNA. Now this is a record of information that is stored and a copy that is almost identical with a few changes that have accumulated during the development of your body is almost identical in every cell in your body. So here there's a slightly cartoonish version of a neuron, but if you were to look at a skin cell or an immune cell from your body, you would find basically the same copy of information. The notable exception, of course, being red blood cells that circulate through your blood and carry iron. Those don't have DNA because they've actually destroyed their nucleus and got rid of it. So they're one of the few cell types in the body that doesn't have a copy of this amazing molecule. So DNA is fundamentally an information-carrying agent, and we can think about that information in multiple different ways. I think that the way that we often think about DNA, of course, is a kind of fingerprint. I think most people here in this room will know that DNA is fundamentally unique to every individual. And in fact, it's safe to say that the DNA strands, the DNA sequence that's present in your cells is completely unique to you. That same sequence has never existed at any other time in any other human being in the history of the world. And in fact, even if you're one of a pair of identical twins, like these 2 cute girls here, it is still the case that your DNA is unique because there are mutations that arise during the process of development, a handful of mutations in the case of identical twins that actually make it possible to tell the difference between these 2 individuals. So even if a pair of identical twins are to leave their DNA at a crime scene, it's actually possible to tell, theoretically at least, from that relatively small number of changes exactly which of the twins committed that particular crime. I don't think that's likely for these 2, however. So DNA is in fact a unique fingerprint, and it's a really big part of what makes us unique and diverse. What differentiates the many different members of the human race and the many different members of the human race that are representative here in the audience from one another. Although, of course, there are many other effects that come from the environment, as I'll emphasize later. So in addition to carrying information about your identity, you can also think of your genome as a kind of history book. And it carries a record of history not really just about you, but also about your ancestors. And I mean your ancestors both recent and also very ancient. So DNA, as far as we know, the DNA that is present in all living creatures arose only once. So to the best of our knowledge, all living things that exist on the face of the planet share a single common ancestor who lived somewhere between 3.5 and 4 billion years ago. And that single common ancestor, through the process of evolution, has given rise to the incredible diversity of life that exists around us. So in this basically almost impossible to parse and very complicated tree of life, we can see just a subset of the many different species that exist and populate this enormous tree of individuals, this enormous tree of species, all of whom are fundamentally related to each other. In case you're curious, humans actually form part of this one little tiny insignificant branch up here, which constitutes the whole of the mammalian species. Most of the species that are found on the surface of the Earth, of course, are tiny little unicellular organisms, incredibly important for everything that we do, but invisible for most of our lives. So that's information that DNA carries about our ancient history. It also carries information about our very recent history, the very recent history of our ancestors. This is a map, and this is a map that has been created entirely using people's DNA. Let me explain to you how this was done. So this is a map that was created by taking individuals who had very specific ancestry. These are people where all 4 of their grandparents were born in the same country. And then several thousand of these individuals were taken. They were genotyped, their genomes were then assayed using a particular genotyping chip that provided information on several 100,000 pieces of particular sites of DNA throughout their genome. And then those individuals were clustered purely based on their genetic relatedness, so how closely related they were to each other, and then projected using a mathematical approach called PCA onto this particular map here. And some of you may already recognize this map. For those of you who don't, the color scheme is exactly the same. These are individuals that were colored purely on the basis of genetic information, and this is the countries where they actually came from, and you can see that there's a stunningly good correlation between this map built entirely based on the genetic information in these individuals and a map that is actually the geographical map of Europe. So people from Spain, most of whose ancestors come from Spain, tend to be more closely related to other Spanish individuals than they are to people from France, for instance. And this pattern of relatedness is something that we can get and although you can see that there's a great continuity between different populations, that means that each of us carries, in effect, a genetic postcode that provides information about the exact positions that our ancestors evolved in. And that will actually come up again later in my talk as I talk about my own DNA. Now, I think there's a popular perception of DNA that is encouraged by the media in many different ways when they talk about a gene for X, a gene for diabetes, or a gene for multiple sclerosis. The DNA constitutes a blueprint, that it sort of pre-specifies everything that we will ever become in our lives. And I think that's really not a constructive analogy to use when thinking about DNA. In fact, what I would suggest is that we think about DNA as a recipe. So DNA specifies, in effect, an ingredients list and a set of instructions that tell our bodies how to put those ingredients together. But that recipe can be interpreted in very different ways depending on the environment that we find ourselves in. So in the same way that the same recipe can give rise to 2 very different outcomes, depending on exactly who's actually conducting that particular experiment, the same genetic recipe can also give rise to very different outcomes. So here I think is one of the best examples I've ever seen. This is a pair of identical twins. They were both born in Ohio. The twin on the left stayed in Ohio. She stayed out of the sun. She never smoked throughout her life. The twin on the right, for whatever reason, decided to move to Florida. She smoked for 40 years. And you can see written, despite the fact that the genomes of these 2 women are basically identical, apart from that handful of spelling errors that I mentioned earlier, you can see the effects of the environment are very clearly stamped across these 2 individuals. So again, these recipes, the same recipes, are interpreted in very different ways in different environmental settings. And over the last 10 to 20 years, using a series of increasingly sophisticated technologies, it's become possible to start to pull apart those recipes, to really understand exactly which pieces of that enormous long stretch of DNA are actually telling our bodies different instructions that are related to, for instance, our risk of diseases or particular traits that are present in us and not necessarily in other people. So I'm not going to go through the full collection of genetic information that we now know about, the collection of information we now know about our genome. But over the last 5 years in particular, we've been able to identify many regions of the genome that are associated with traits like obesity, We found regions of the genome that affect our response to drugs. So for instance, it now becomes possible to predict for many drugs exactly who will respond to those drugs and who will have an adverse reaction to those drugs. DNA can predict actually with considerable certainty now the color of a person's eyes, the color of their hair, and the color of their skin. That turns out to be relatively easily genetically decoded, although it's not perfect in everyone. And there are also many other more complex traits. So for instance, some of you may be aware of this, but there is a subset of people in this room who are capable of knowing, by smelling someone's urine, whether or not they've eaten asparagus in the last few hours. So does anyone in this room know what I'm talking about? Does anyone have any? Okay, right, great. So me as well. So it turns out, and you look at, as you see those hands that went up around this room, it turns out there is a subset of the people in this room that will absolutely unambiguously know what I'm talking about because they can smell asparagus on their own urine if they've eaten it. There's a subset of these people in this room who are looking at their neighbour in complete incomprehension because they've never realised that such a thing was possible. But I assure you that is the case. And that turns out that a lot of the difference between individuals in that particular trait turns out to lie in a region that encodes olfactory receptors that encode our sense of smell. And so that appears to at least partly explain the difference between different individuals in the population in response to that particular trait. But despite the advances over the last 5 years, there are still many pieces of the genome that we really don't understand exactly what they're doing, and many diseases where we are far from fully understanding all of the genetic contributions to those diseases. And that's actually particularly true in the diseases that I care about the most. These are very rare, severe diseases such as muscular dystrophy and congenital myopathy, diseases that often take effect very early in life, that have very severe effects on survival and on people's fitness, and I'll show some examples of them later. And where, although for many cases we can now identify exactly which genetic change has caused that particular disease, there are many other cases in the general population for whom we cannot yet find a cause. And so that will be the focus really of the second and third parts of my talk. Before I get to that though, I wanted to take a little detour and I think illustrate some of the bits of information that we can get from our genome using a couple of nuggets of information that I've managed to glean from my own genome. And this is primarily through, I've actually had a number of genetic tests performed on myself over the course of the last 7 years now. And most of the results that I'm going to be showing today are from a relatively cheap test, a $100 test that I purchased from the company 23andMe. So 23andMe, for those of you who don't know, is one of multiple companies that allows you to actually order a genetic test without the input from a doctor. And they can then provide, you spit into a tube, it takes about 15 minutes, you have to fill up 2 mils of saliva in a tube. That's actually, it turns out, a shockingly large amount of saliva to produce, as those of you will know. Once you've sent off your saliva, 4 to 6 weeks later, you get a set of results that you can look up online that gives you all sorts of information about yourself. Some of you may know of 23andMe because, of course, there was a controversy a couple of years ago now where parts of the company were shut down by the FDA because they were offering health advice without providing a doctor's assistance. They're now slowly getting FDA approval to start that part of the company back up again. I'm not going to talk really about disease risk at all in this presentation. I just wanted to focus mostly on relatedness and ancestry. So these three maps are maps these three maps of my genome actually are maps that are provided by 23andMe, and each of them shows, here we have each of these lines here is a different chromosome. One of my 23 pairs of chromosomes, down here is an X chromosome and a Y chromosome. I'm a boy, so I carry a Y. And what these maps show is how closely related I am to another individual who also happens to be present in 23andMe's database. In light blue are the sections of my genome where I am 50% related to that individual. That is, one copy of my DNA is shared with that other person, but the other copy is not. In dark blue are the places where I am 100% related to that other individual. And in white over here are the places where I am completely unrelated to that individual. That is, despite the fact that all humans are fundamentally related, that means that 23andMe's algorithms were unable to detect any recent relatedness between me and this other person. So these are 3 people that I know actually extremely well. The first person is my father, and I was very relieved to find out that I am in fact 50% related to my father at every position in my genome. That was relieving both for me and I think for my father, possibly my mother as well. The second person is my brother, and as brothers are, as siblings are, it turns out that I am not related in some parts of my genome. I am 50% related in others and 100% related in others. And this has to do with the random shuffling of our DNA during the formation of sperm and eggs. It turns out I'm actually slightly more than 50% related to my brother. So it turns out I'm slightly more related to my brother than either of us are to our parents, which I think really brought us closer together as a family. That was fantastic to find out. And the third person with whom it turns out I'm not related at all turns out to be my wife, who is here in the audience. And so again, this came as an enormous relief to find out that we were not in fact undiscovered distant cousins. So that was amazingly relieving. So we can find other information, of course, out about our genomes. Here is a map that's very similar to the map that I showed earlier, although not quite as pretty, I must admit. This is a principal component analysis of individuals in the 23andMe database. Here showing people of known genetic ancestry. And this is not just a map of Europe, this is actually a map of the world. So there are sections out here that correspond to East Asia and the Pacific. This section down here corresponds to Africa. And this section up here corresponds to people of European ancestry. This little green blob here is me. It turns out, and I think this will come as no surprise to those of you who've been listening to me, that I am generally of European ancestry, in fact, almost exclusively as it turns out. Most of my genome comes from the UK. But I was surprised to find out that despite the fact that there is a very large fraction of my genome that is clearly of British ancestry, exactly as expected from my oral family history, there were 2 stretches of DNA shown here that were actually very confidently assigned as being of South Asian ancestry. So that was a real puzzle. There was actually no recorded family history that we were aware of that indicated any evidence at all of South Asian ancestry in my family. My brother, who'd also been tested, also turned out to have a slightly different set of South Asian ancestry. We shared 1 block, but not others. And my father had even more. So it turns out we could tell from this actually very clearly that we had inherited these blocks of South Asian ancestry from my father. So it must have been a South Asian ancestor on my father's side. So I'm not going to go through in great detail the forensic analysis that then proceeded as we dug through our family tree, spent a lot of time digging through genealogical records to try to figure out where this had come from. And it turns out we were able to demonstrate with some certainty it actually came from this man here, my great-great-great-grandfather, Captain Abel Henry Chapman, who was born in England, but as it turns out spent 15 years in the British Army in India. We don't know much about his time there, but we know that he met a woman called Molly Fraser. Molly Fraser was born in Calcutta. We know even less about her, except that her father was English. We know absolutely nothing at all about her mother. And Abel and Molly got married. They then moved back to England together. They started the Chapman family that eventually led to my father's lineage. And we are almost certain, based on tracing this lineage, that Molly Fraser was in fact 50% Indian, that her mother was in fact Indian. And this was something that my family had absolutely no idea about. My grandmother, who would have been absolutely shocked to hear this, by the way, had actually passed away about 5 years before we learned this. But the rest of the family was extremely excited to learn that we actually have these little chunks of our genome that give us information not just potentially about our health risk, but also tell us about a love story that occurred more than 150 years ago on a different continent, which is kind of cool. So that's all I'm going to say about my genome. I wanted to spend the rest of the presentation focusing on the ways in which the same types of information that we can get in the context of these tests and the same types of information that are carried in all of our genomes can be used to learn more about the genetic basis of rare diseases. The diseases that my lab study have tended to study over the last 4 years are diseases that affect muscles. These are 2 girls from a family. This is actually one of the first families we ever studied in my lab. Both of these girls, and these are actually the only 2 children in this family, are affected by a disease called Mendelian myopathy. Mendelian myopathy is present from birth. You can see in this younger girl here that she was born actually quite weak. It affects all of the muscles in the body to a greater or lesser extent, but you can see in both of these girls here, the most obvious and visible manifestation of the disease is an extreme weakness in the facial muscles. And in particular, their jaw muscles are so weak that they can't close their mouths. So their jaws basically hang open. So this disease is obviously very debilitating. Despite this, the older girl has learned how to walk with support, and she's also learned how to talk, which is amazing. She's learned how to use her hand to move her mouth up and down and actually communicate. So she's dealt extremely well with the challenges that her family faces. In addition to those medical challenges that they face, the family, and at the time, the oldest girl here is 9 years old. For those 9 years, this family had another challenge to deal with, and that was the extreme uncertainty surrounding the exact cause of the disease that was present in both of their girls. So at the time that the DNA from this family was first sent to us, they had gone through 9 years of what we call a diagnostic odyssey. And this is actually a pretty characteristic length of time for a rare disease patient to have spent searching for a genetic cause for their particular disease. I won't go through the details here, but both girls had a muscle biopsy taken. That's a standard diagnostic procedure. It's a painful procedure, and that's used to actually stain for a whole series of different proteins within the muscle that can help figure out what gene might be disrupted. A whole series of genes that were known to be involved in Mendelian myopathy had been scanned. No results were found. And 9 years later, the family was no closer than they ever were to learning exactly what the cause of their children's disease was. And that's not a rare story. In fact, this turns out to be true for about half of all rare muscle disease patients. They can't be given the precise gene that actually causes their child's disease, using at least using standard diagnostic techniques. So we received the DNA in the lab here at the Broad. We were then able to send that DNA off for a process called exome sequencing. That word's going to come up a bit during the rest of my talk. Exome sequencing is a method that allows us to perform sequencing, to extract the sequence of DNA just for the 2% of the genome that codes for proteins. So this is the parts of the genome that we understand the best, and these are also the parts of the genome that tend to be extremely enriched for the genetic changes that cause severe diseases like congenital myopathy. And when we sequenced the DNA of these two girls and both of their parents, what we found was that both girls carried two different changes within a gene called LMOD3 or lyomodin-3. There's this insignificant little change here, a change to an A, and there's 3 bases that are deleted down here. Now it turns out this change up here creates a stop codon in this gene, and that's basically a signal that tells that gene to stop producing protein. So this is a completely non-functional copy of this gene. This copy down here actually results in a protein that is missing one part of the protein, one small amino acid or section of that protein. And these two changes in combination, these two tiny changes among the 3 billion letters of DNA that I showed you earlier, were responsible for causing this catastrophic disease in this family. This is true for this is of course true for most rare diseases. These are almost all what we call monogenic diseases. That means that a handful of changes, 1, maybe 2 changes within that particular gene, are sufficient to cause these very severe diseases that profoundly affect their ability to survive and to thrive. Now, a diagnosis in these girls did not profoundly change their outcome, but it did have a number of impacts on their day-to-day life. This is true for rare disease patients in general, actually. It is true for a small handful of patients, a small but growing handful of patients, that we can actually use a diagnosis to lead to a change in therapy. So we've now sequenced in our lab more than 1,000 families affected by rare disease. For more than 400 of those families, we've been able to give them back a diagnosis. And for several dozen of those families, it's been possible then to convert what had previously been effectively an untreatable disease into a disease where it is actually possible to give medication to alleviate at least some of those symptoms. Now that, of course, that small fraction will of course grow as we begin to develop more and better drugs to treat these rare diseases. A second advantage is that these patients learn more about their prognosis, so they can predict better exactly what the effects of their disease will be over time. For many of the families, this allows them to do reproductive planning, so they can come up with ways of ensuring that future children that are born within that family do not suffer from the same diseases, and that means that often they can actually go on and have children, whereas they were planning to not do that before. And then finally, and I actually think this point is really critical, it becomes possible to actually put a name to a disease. And for families like the one that I showed earlier, you can imagine after 9 years of hunting for the cause for a child's disease, it becomes as an enormous relief to actually have a specific cause, a specific gene, and a specific name that you can give to that disease, and also to be able to find other patients who have the same diseases. So in the case of the patients that we diagnosed with LMOD3 mutations, over the course of the next 4 to 6 months, we were able to find 12 other families around the world that had mutations in the same gene that gave rise to Mendelian myopathy. That means it becomes possible to then form networks of people who understand your disease and also who can start to lobby for both pharma companies and governments for potentially more effective therapies for those diseases. So what can we do to improve the diagnosis of patients with rare diseases? One of the things that we have found extremely useful in our lab, and this is true from the very earliest days of exome sequencing in my lab, is that what we need to be able to do is to take all of the genetic changes that we find in our patients and compare them to extremely large databases of normal variation throughout the population. And this is important, I think, what we need to be able to do is to answer 3 very simple questions about that genetic variation. We need to know for each change that's found in our patient, has that ever been seen before in anyone who's ever been sequenced? If so, how common is it in the general population and across different populations? And is that frequency within the population consistent with a rare disease-causing mutation? And it turns out that the frequency within the population is one of the best clues we have as to whether a variant could actually be a cause of a specific rare disease. The final question, of course, is that if there are other individuals out in this population who carry that particular variant, we'd also like to know if they also have the disease or if they have the same combination of variants if they also have that disease. And again, that's a really important clue as to finding out if that particular genetic variation could be responsible for that particular patient's disease. Now, we live in an absolutely remarkable time to be doing this type of human genetics. And one of the reasons for that is that there is the sheer scale of the data that's now been generated around the world on the DNA of now more than 1 million people who've had either their exomes or genome sequenced. So this is a tremendous amount of information that potentially provides huge amounts of data on the distribution of genetic variation across the general population. The challenge, of course, is that most of that information is inaccessible for several reasons. So some of that information is generated by companies and by academic groups. Some of the companies regard that information as being proprietary knowledge. You'll be shocked to learn perhaps that some academics also are not keen on sharing their particular pieces of data. There are also mundane obstacles that prevent data sharing. It turns out to be very difficult to move large amounts of data from one place to another. And another final problem is the fact that different projects tend to generate their data in different ways. And so because each project tends to use their own analysis pipeline, it becomes very difficult to take the end results of all of those pipelines and merge them all together without being completely, hopelessly confounded by technical differences between those different pipelines. So around about 3 years ago, we set about trying to solve that through an exercise that came to be known as the Exome Aggregation Consortium. And to explain what that is, I'm actually going to explain what each of the words in the title means. We've already talked about exome sequencing, so that is a technology that is now being used at very large scale that provides a cost-effective way of understanding the sequence of just the protein coding pieces of the human genome. And this has been applied now to hundreds of thousands of people around the world. The term aggregation refers to the fact that in this particular consortium, our goal was to take raw data from many different projects and merge that all together into a single unified project. It's very inspiring, thank you. And finally, the word consortium refers to the fact that no one research group can actually do this alone. It was absolutely essential that we could work with groups around the world who had generated this exome sequencing data from tens of thousands of people and pull all of that data together into a single unified resource. So I'm not going to go through the details of the process by which we collected these data. I will instead go through the headline numbers here. So we were able, by the middle of October 2014, thanks to the help of a number of consortia who I'll mention at the end of this talk, to pull together the raw sequencing data from over 90,000 people. And this is a staggering amount of data, as I'll show in a second. These were donated by 21 different research studies, most of which were focused on understanding the genetics of common complex diseases, such as type 2 diabetes and schizophrenia. These were all processed through exactly the same pipeline, and this required an enormous amount of computing capacity for which we were totally dependent on the raw computing power that's present here at the Broad Institute. This would have been extremely difficult to do almost anywhere else in the world. We were able, at the end of that process, that enormous and painful process, to call genetic variants, that is sequence changes, across all 92,000 samples. And because of this process known as joint calling, this simultaneous analysis across all of these samples, we could be sure that the same genetic change had been called in exactly the same way across all of those individuals, so that we had very accurate estimates of frequency for every variant that we discovered. At the end of that process, we then filtered it down to just 60,000 samples. We removed about a third of the samples for good reason. We removed them either because they had low quality data, or because they were related to one another, or because they or their first degree relatives suffered from a very severe rare disease. We wanted this to be as representative of the general population as possible. So this is designed to be a control data set. The final data set composes over 10 million genetic variants. And these variants are almost all new. They'd actually never been reported before. We were the first group to discover them in aggregate in this particular project. And this is really the first time that we were able to peer down at variants that were present at a frequency of less than 1 in 10,000 and even less than 1 in 100,000 within the population. And at the end of this process, we were then able to release that frequency data to the world in a website that I'll show in a couple of slides' time. So just to emphasize the sheer amount of data that we had to process here, this is about a petabyte of raw data. That's 1,000 terabytes. So that would be equivalent to taking a cutting-edge MacBook Air and then actually stacking up 4,000 of those laptops and copying 1/4000th of the data onto each of those laptops, which is an exercise I actually really strongly don't recommend. We were fortunate enough, though, to be able to have access to the Broad's computational cluster, which is more than capable of actually storing this petabyte. In fact, it currently stores 10 times that amount of data. So this is what the data look like when you've called variants across 60,000 people. This is a principal component analysis plot. I've already talked about PCA a couple of times during the talk. The only difference here is that this is 3D and fancy and spinning, thanks to Michael Leck in the audience. What this shows us is the patterns of relationship between the samples that we sequenced in the exact data set. In blue, we have individuals of European ancestry, and actually more than 50% of the samples in our data set are European, and that's a bias in the ascertainment of the studies that contributed to the sample, to this data set. But you can also see we have thousands of individuals in this data set from Latino samples, South Asian, East Asian, as well as African samples. So this is by no means a comprehensive sampling of human genetic diversity. You can see, for instance, East Asia is sitting all off on its lonesome down here, and that is because we're missing huge swathes of genetic diversity through Central Asia and the Middle East that would normally be connecting East Asia right the way through up to Europe. So it's not a perfect representation of human genetic diversity, but it does mean for many of these populations for the first time, we have very deep ascertainment of frequency data that we can use to study rare disease patients from each of these different populations. And that data is available to the world on a website, exact.broadinstitute.org. If you have a favorite gene, you can look up your favorite gene on this website. It will give you information about how much data we have for each of the pieces of that gene. And also down below that, we'll list every single variant that's been discovered there. You can click on those variants to learn more about its frequency in the general population and across multiple different populations. So just to illustrate how EXACT can be useful, let's go back to our family, family A, and those 2 mutations that we discovered in the LMOD3 gene. Now, it turns out both of these variants are extremely rare. This stop codon up here was seen only once in 60,000 people in EXACT. So because every person has 2 copies of the LMOD3 gene, that means it has a frequency of less than 1 in 100,000. And this deletion down here is actually never seen in the EXACT database. And in fact, the LMOD3 gene is the only gene in the genome where 2 rare protein-altering variants of this frequency were found in both of the affected children in this particular family. So the EXACT dataset allows us to zoom in almost with complete precision in the causal gene in this particular family. And that turns out to be true for many rare disease patients. In this plot, we're basically just showing the efficiency of filtering of variants found in rare disease patients using a previous publicly available dataset of exome variation. This is called ESP, and our dataset, EXAC. And what you can see is that EXAC dramatically reduces the number of candidate variants that are present in a patient's genome from a whole range of different populations, simply because it is so much larger and so much more diverse than previous publicly available datasets. And this has had an impact not just in our lab, but in labs around the world. So to date, the EXAC website has had over 5 million page views. You can actually see up here the pattern of page views over time. You can see that people don't tend to search the EXAC website on weekends, which is very disappointing. They take a break. They take a break at Christmas here as well. Virtually all countries have actually had hits on the EXAC website, although we're still missing hits from Togo as well as from Svalbard, and we're still pushing for people to actually visit those locations and log a hit on the EXAC website. But it's been gratifying to see this resource used so heavily and by so many different groups around the world. And now we estimate to have made tens of thousands of diagnoses based on the information that's contained here. So that's the information that we can use. That's the way in which we can use this EXAC dataset to understand the causes of rare diseases. There are many other scientific applications of this dataset. I'm not going to have time to go through those in detail here. I'll just mention them very briefly. We can explore the patterns of mutation and of variation across all human genes and learn in a much higher resolution exactly how those patterns vary from person to person and across populations. Critically, and this has actually turned out to be a very exciting area of research, we can look not only at the variants that are present in the EXAC dataset, but also at the variants that are missing. And this is work that's been led by Caitlin Samoka, a graduate student in Mark Daly's lab, working with our team. Caitlin has developed a model that allows us to predict, actually with quite a great degree of precision, which pieces of the genome have less variation than we would expect to see by chance. And that, of course, points us to regions where variation is missing because it tends to be damaging when it's found in the population. And it turns out these regions are incredibly enriched for disease-causing mutations. So this map of missing variation actually points us to regions in the genome where if we find a variant in a disease patient, it has a much higher probability of being disease-causing. And that's already proving useful for our rare disease patients, as well as for studies in autism, schizophrenia, and other diseases. And a final use, and again, I won't have time to talk about this in detail, is finding what we call human knockouts. So I think some of you may be familiar with the concept of a knockout mouse, which is a mouse that's been engineered to lack a particular gene. So a specific gene has been knocked out in that mouse. And knockout mice have been generated now for more than 20 years. They're extremely useful in understanding exactly what the function of those knocked out genes are. For fairly obvious ethical reasons, it's not possible to knock out genes in humans. But of course, we can look for rare individuals who have knocked out those genes through naturally occurring variation. In other words, where both copies of that gene have actually undergone a mutation at some point during their evolutionary history that results in those genes being completely inactivated. There are, in fact, more than 1,000 genes where at least one individual within the EXAC dataset shows a signature of being knocked out in at least one person. So we're now studying those human knockouts to see if we can learn more about the genes that are affected by those mutations. Now, we have a lot of work ahead of us with EXAC, and of course with many of the projects related to EXAC around the world. And these fall, I think, into a number of different categories that we're pursuing, at least over the course of the next year. One big challenge ahead of us is simply increasing the scale of the data. Now, it turns out we can do a lot of good with 60,000 exomes, but we can do even more good as we start to increase that number of samples. And in particular, when it comes to identifying regions of the genome where variation is missing, the resolution of those regions of the genome becomes better and better as we add more and more samples to this data set. So later this year, we're hoping to be able to release a new data set that will have twice the number of samples. So have more than 120,000 samples, giving us completely unprecedented resolution into those regions of the genome. Now, up until now, of course, given the nature of exome sequencing, we've been focused on protein coding bits of the genome. So that is the 2% of the genome that actually encodes for protein sequences. We're very interested in moving beyond that, and later this year, we'll be releasing a version of EXAC that looks at the whole genome for more than 10,000 samples and actually allows us to look at the patterns of variation in other bits of the genome as well. And finally, and this is in a series of relatively small-scale pilot studies, we're starting to think about ways in which it might be possible to find individuals who have what we call extreme genomes within EXAC. So these include people who have those knockout variants that I mentioned earlier, or who have variants that would be expected to cause very severe disease, but as far as we can tell, the person doesn't actually have that disease. We can take people with these extreme genotypes and potentially, although EXAC was never designed to do this, in some cases it turns out to actually be possible to recontact those individuals, learn more about their health status, and potentially understand exactly what that particular gene is doing, or why they are actually protected from that particular disease. And we're very excited about those studies as well. The move of genomics into big data, of course, though, will continue, and this will continue in EXAC and in many other projects around the world. And I think I actually want to finish on this point, that it's worth noting that I think genomics in some ways and genetics in general has a little bit of an inferiority complex. Sometimes we think about genetics as being kind of big data, but I'd actually argue that, and this is a slide that's contributed by Eric Banks from the Data Sciences platform here, but I'd actually argue that human genomics has already reached the stage of big data, even in comparison to major technology companies. So one genome is, given that we sequence every genome to high coverage, so that is each base in the genome is sequenced multiple times, it turns out the amount of data that we produce when we sequence a human genome is about 120 gigabytes of data. So by comparison to that, the average Netflix user streams about a third of that every month in watching their favorite shows. So your human genome is worth about 3 months worth of the average Netflix user's data. Now, it turns out the Broad sequences about one of those genomes every 10 minutes. And this is, I don't know how much you guys know about the Broad sequencing facility. It's located about 4 blocks that way, and it is a factory for churning out data. It is actually the biggest factory in the US for producing DNA sequencing data, and it does so at a staggering and, in fact, at an intimidating rate. So that means that given that we sequence one genome every 10 minutes, the Broad produces more information than the entire Netflix library every 2 months. And that pace of generation is continuing to grow. So right now we store 10 petabytes of genomic sequence data in our data cluster, and that is a phenomenal amount of data, even compared to something like Facebook. So Facebook stores 10 billion photos in their database, and that takes up only 1.5 petabytes of space. So that's only 15% of the amount of data. So all of the photos that you've ever seen on Facebook, and in fact all of the photos that anyone has ever seen on Facebook, take up just 15% of the data that the Broad has stored from our genome and exome sequencing activities over the last 10 years. So we have already reached the whole genome era, and I think we have reached the point where this large data is really starting to have an impact, not just on rare disease diagnosis, but on understanding the biology of every gene in the human genome. So with that, I'd like to finish by thanking everyone who's been involved in generating these data. I actually particularly wanted to point out a few people here. Monkul Lek is a postdoctoral scientist in my lab who was involved not just in the EXAC project, where he was the lead analyst, but also in many of the rare disease projects that are being conducted in the lab, including the diagnosis of the LMOD3 family that I mentioned. Eric Minikel, Konrad Kaczewski, and Caitlin Samoka all contributed very actively to the work in the Exome Aggregation Consortium, as well as many other members of my lab and other labs. I'd like to thank all of the principal investigators who agreed to donate their data to the EXAC project. Without these people's willingness, not just willingness to donate data, but their enthusiasm about allowing the data that they had generated, this is approximately $50 million worth of data, with allowing the data that they had generated to be pulled together for the common good, projects like EXAC would simply be impossible. And I also wanted to finish by saying a huge word of thanks to two groups of people here at the Broad. The Broad Genomics platform, which generated more than 90% of the sequencing data that was used in the EXAC project, and also a huge thanks to the Data Sciences platform here at the Broad, which produced the tools that were absolutely required for us to perform, the tools and the computing power that were absolutely required to perform an analysis on the scale of EXAC. With that, I'd like to finish by thanking you guys for listening. Thanks very much.
Hi, thank you for sharing your knowledge. My question is, today we know how to sequence our DNA. Do you think that in the future there will be a potential that we have frequent access to our own genome in a way that we would be able to predict or to see actually any mutation that can happen through the course of life that could lead in a disease?
Sure. So I think there's two questions in there. One is about whether things will move in the direction of people having better access to their own genetic data. I think there's strong evidence that in fact the world is already moving in that direction. So I mentioned there's already companies out there that make it possible to get access to at least some small fraction of your genome in a direct-to-consumer fashion without necessarily having to go through a doctor and get direct access to your genetic information. So that's already happening. There's a lot of work still to do though, and I think there is still large sections of medicine that are still stuck in a more paternalistic frame of mind where there are various obstacles that are put in the path of people who want to learn more about their DNA. The second part of your question though was about changes in DNA that occur during life. And although it is true that all of us are born with a genome, a particular genome that is present in the fertilized embryo that gives rise to all the tissues in our body, over the course of our life, of course, each of our cells undergoes mutations. Some of those mutations are very important because they can potentially lead to cancer. Cancer is fundamentally a disease that is caused by mutations that have occurred in those cells. And I think the answer is yes. It's already becoming the case that when people have cancer, when they're actual cancer patients, genome sequencing is now almost routinely applied to the tumor and to the blood DNA from those individuals to learn more about the exact genetic changes that have occurred in that cancer and potentially to tailor therapies to those cancers. And a lot of that work is actually being done here at the cancer group here at the Broad. But it will also be possible to sequence the blood from an individual and track the presence of mutations over time that may indicate the presence of a pre-malignant cancer in their bloodstream. I think we're still some way towards reaching the point where we can actually apply that as a screening test. There are companies that are thinking about doing this. There's a company called Grail that's been started on the West Coast to do this. I think that we're a long way away from having a perfect screening test because the false positive rate is still too high. But ultimately, you can imagine as we increase our resolution and our database of information from much larger numbers of individuals, it will become possible to do that, for sure.
Thank you.
Could you comment on the story in the news about two or three days ago involving, I think, some part of the United States government planning a project calling for 1 million volunteers to make available their genomes. Thank you.
That's a relief, actually. I thought you were going to ask me about Melania Trump's plagiarism scandal. So it's like, I can't say much about that, but I can certainly talk about the initiative you spoke about. So this is also known as the Precision Medicine Initiative. This is an NIH-funded and in fact, White House-supported initiative that's just been launched. I think this is an incredibly exciting project. The long-term goal of this is to recruit at least 1 million, and in fact, the total number will likely be much higher than 1 million volunteers from around the US. This will happen in a number of ways. There will be engagement with health systems, so people who are involved in particular health systems around the US will be recruited into this process. It will also be possible to directly volunteer for this project, so anyone here in the US can actually volunteer and sign up to become part of that project. The long-term vision for what will actually happen with those projects is not yet fully determined, but what seems likely is that the project will collect information not just about the genetics of those people, hopefully eventually including whole genome sequencing, but also lots of information about their electronic health records, as well as questionnaire information and all sorts of other information about their environment and their health and their traits. And that will then be combined together in a way that's actually made accessible to researchers. Now, I think there are obviously privacy concerns that have to be addressed by this project, and I think that those will be raised and discussed in many ways over the coming months. But I think it's worth emphasizing just how tremendously exciting this project is. With 1 million people for whom we have complete genetic data, as well as a huge amounts of information about their environmental risk and their health status, it will be possible to reveal information about the genes that cause disease that were previously completely inaccessible. It will be possible, for instance, to find the human knockouts that I mentioned earlier among those 1 million people and determine exactly what effects those gene knockouts have on health. And it will be possible to study all sorts of things that actually we really have no idea about right now that will emerge completely unexpected from a data set of that magnitude. So I think all of us are actually extremely excited about seeing that project come together. We hope that it persists beyond the current election cycle and that there is a strong long-term funding commitment to that project. And I think that really will transform the way that we think about genomic medicine.
It's a great talk. Your last few slides made it pretty evident as the amount of data that's available. what specifically would be required for the analysis bit of it, not just the hardware, but the methods that we used? I mean, analyzing 1 million data sets is completely different than doing just 1,000 data sets.
Yeah, that's a great question, and it's actually a question that I think keeps a lot of us awake at night. So I didn't actually show this sort of growth curve. I just gave you some sort of absolute numbers of the number of genomes that the Broad produces. It's worth emphasizing that the rate at which that growth, at which that data is going up is just staggering. It's basically an exponential increase in the amount of data that we generate per day. So that means we have to fundamentally change the way that we run our analyses. The analysis methods that worked a year ago or 2 years ago start to break down very quickly as we start applying those to 10 times or 100 times as much data as what we had back then. And so there's, the Broad is at the forefront actually in many ways in leading the development of methods that can be applied across these very large scale genome databases. And there's a couple of things that I can mention very briefly that I think will be really important here. The first is that we here at the Broad and many others around the world have started to move our data onto the cloud. So most of you I think will have heard of the cloud. The cloud is basically a collection of computers that exist somewhere else other than here. And the cloud is extremely important because it means that it's possible to actually use it in a very flexible way. So rather than buying a certain number of hard drives and storing your data on those hard drives, it becomes possible to rapidly change the number of hard drives that you have access to spin up a new set of hard drives and store more data, perform a particular analysis, and then move that data off and then close that down. So that increases the flexibility that we have for data analysis. The second thing is that we need to really change the way that we run analysis and the software that we actually run on these data sets. And that involves things like massive parallelization. So technologies, I think, like Spark, for instance, that allow us to do analyses in many different thousands of CPUs simultaneously and spread these genomic data over many different computers and run those analyses on many different computers simultaneously, and then pull that information together at the end into a final data set will absolutely be required for the next wave of genome sequencing. Beyond that, where we are in even 2 years' time, I think none of us really know. I think we're barely going to be able to keep up for that 2 years, but I hope that we can at least keep ourselves afloat just for long enough that we can get to the next phase of genome analysis.
Very interesting talk. I'm curious, what sort of biochemical or biophysical follow-up do you have for these proteins that you find to have mutations? Do you contact a lab if they're working on that family of proteins, for example, or what sorts of things do you do?
Yeah, that's another great question. So particularly in the rare disease setting, this of course becomes extremely important. And this was I think illustrated pretty well in the case of the LMOD3 gene that we found in that particular family. So that gene at the time, there was very little known about it. We knew that there'd been a little bit of biochemical work done showing that it interacted with a set of other proteins, some of which were also known to also be involved in that particular disease, but not much else had been done. So over the course of actually the next 2 years after the discovery of that gene, a small army of researchers was actually mobilized by one of our collaborators, Nigel Clark, who basically pulled a consortium of collaborators together with expertise in different areas, including zebrafish modeling of a particular gene. There was work on a knockout mouse of that particular gene. Biophysical analyses were done to look at the binding of this particular protein to actin in a sort of disaggregated state. And a ton of different cell biology experiments were also done simultaneously, all of which pointed to a very specific role for this particular protein in the same complex that was known to be involved in other proteins that cause the same disease. Now that was an extremely labor-intensive activity. I think that the key question that we need to think about, and in fact a question that exercises a lot of the minds here at the Broad, is how do we scale that up? How do we take that type of gene-by-gene activity and apply it to the level of discovery that we're likely to be seeing over the next few years? I think there's a couple of ways in which we can go about doing that. One is developing these very flexible assays like RNA sequencing and protein analysis that we can actually apply to lots of proteins regardless of what their function is. A second thing is to really think hard about what the core functions are of lots of proteins across the genome and develop robust assays that can be used across lots of different genes simultaneously. And then I think there's lots of other things that we just haven't yet thought of, robust, high-throughput assays that can be applied in many different ways to learn more about the function of a gene that we discover as we start applying these high-throughput technologies.
So to me, so there seems like there's a discrepancy between how much data we have and how much information we process from the data. So for example, you said only 15% of the data constitutes of all the photos like pretty much everyone has ever taken. And with all those photos, you could pretty much reconstruct like everyone's lives and everything about their lives. So is it because like the genomic data is like so, I guess, low quality, or have our tools not caught up with how much data we actually have?
So I think fundamentally it's a question about how much useful information is present in the Facebook photo library compared to the genomic library. That's an interesting question. So first I'd quibble, I think, with the idea that the Facebook photos could actually be used to reconstruct the entirety of people's lives. In fact, we know that Facebook in general actually only shows us a very specific sliver of people's lives that they choose to display. But nonetheless, the photos have an amazingly high information content, and I think that's true because the human brain is capable of processing images and to glean both factual and emotional information from images in a way that's tougher from digital information. The core part of your question, I think, related to the quality of the underlying data, and I think there is a lot to be improved there. I mentioned just very briefly in passing that at the moment, we don't just sequence a genome once when we sequence it. We actually have to sequence it somewhere between 30 and 40 times to build up enough information to be very confident that a particular region of the genome actually has a variation or not. And that is really a reflection of the fact that the underlying data is still noisy. We have very short segments of DNA that we have to map back to the genome, and those little segments of DNA have a relatively high error rate. So we have to increase the amount of data that we generate to really be confident that we've actually called a variant properly. As technology increases, the requirements for that DNA data will actually start to go down. The second thing is that 10 petabytes of data represents the rawest form of data. So that is the data in the terms of the raw reads that we store. The data that we tend to share with our collaborators is actually a very condensed version of the data that only shows the sites that contain a variation in at least one individual, and that's much, much smaller. So for the exact data set, for instance, in that particular case, we had a petabyte of raw sequencing data, and that condenses down to about 3.3 terabytes of variant call data. And so we can actually store the DNA, we lose a little bit of information in that process, but we can actually store useful information about DNA in a much smaller and more efficient way. There's still a lot that we have to learn, but I think we are getting there in terms of extracting function from this data. Hi.
Hi, thank you very much for your talk. So I'm an undergraduate at UMass Boston, so I might not be quite well aware of the technicality of your presentation, but my question was to do with reference to precision medicine. So I do understand that sequencing DNA and being able to have then 120 gigabytes of data on a person would be very important in terms of being able to treat each individual and being able to distinguish variabilities in different people. However, my question is, so hypothetically, if you have a patient who had a type of malignant cancer and it got treated and therefore the mutations got deleted and you sequence it after it, right? How would you be able to backtrack and find the data in order to be able to be comparable to a different person? And is there any other mode of substitution for the information which would be able to improve how we're able to read a person's DNA?
Right, so I mean, I think in the specific sense of someone who has a cancer, who undergoes therapy and then has a particular mutation that disappears, that's a really interesting question and one that's being actively explored by many researchers. So this is basically, this has to do with a cancer's susceptibility or sensitivity to particular drugs and their response to therapy. Now, of course, once a mutation's disappeared from a cancer, you can't recover it. It's now gone. But there are now a number of projects here in Boston, many other places around the world, where people are sequencing people's cancer at multiple times over the course of their therapy. And that actually allows them to reconstruct the exact genetic changes that occur within that cancer, both as the cancer grows and metastasizes and changes, and also as the cancer responds to therapy. So they can see, for instance, which genetic changes disappear when you target that particular cancer, and which genetic changes actually become more common when you target that cancer. Those genetic changes that become more common are probably genetic changes that confer resistance to the therapy. And so that actually provides really useful information in terms of understanding what the mechanisms are for a cancer developing resistance to a particular drug. That type of resolution, the challenge at the moment, I think, is that it's still expensive to generate sequencing data on a tumor. But as the price of sequencing goes down, it's in the couple of $1,000s per genome right now. As that starts to go down, you can imagine it would actually become possible to sample people's blood or their tumors at very high resolution, so many different times over the course of their cancer, and develop a much higher resolution sense of exactly what's going on, how that cancer behaves over time, and also potentially to tailor therapies as you see a new clone of cancer start to rise in frequency within that person's tumor to apply new therapies that actually target that particular set of cancer cells.
question. I missed last week's talk, unfortunately, on epigenetics. Maybe you could just clarify something for me. When we're sequencing a genome, we're not sequencing the genome in a context, for example, in an epigenetic context. And what we're looking at is we're trying to correlate between the genome and the expression down the road, which we're calling a disease or whatever we want to call it. We call it an abnormality. It may or may not be as such, really. But we're missing all this piece in between, in the middle, many pieces, but the epigenetic piece. And so what I'm curious about is, as we go through and we're sequencing the genome, are we developing techniques that actually identify for this genome that there are, as an example, epigenetic switches there that are not necessarily present on other genomes? Are we beginning to try to look at the bigger picture, as it were, to understand the genome as a whole? Or are we just too early at this stage to be able to do that?
I mean, I think it's a challenging area. I mean, basically, the way, when I talk about sequencing a genome here, you're absolutely right. This is a genome that is stripped of context. Basically, in practice, what this means is we take blood from an individual, the DNA from many cells is then extracted from that blood, and all of that DNA is basically mixed together, independent of cell context, smashed up into little pieces, sequenced, and then stitched back together. So we lose all of the information about the epigenetic state of that tissue as a whole, but we also definitely lose any information about the epigenetic state of individual cells within that person's blood. And that is, as we know, extremely useful information. So there are ways that we can imagine trying to recover that type of information. I think last week you will have heard about technologies that allow us to look at epigenetics on bulk tissue samples at a very large scale. It's also possible actually to start looking at single cells, and this is work that's tremendously exciting actually. It turns out it's now possible to take a blood sample or a tissue sample from anyone, to disaggregate that tissue sample into individual cells, and then to sequence, albeit at relatively low coverage, the DNA and the RNA from each of those cells individually. And so that allows us to actually peer into the impact those variants have at a cell level. So we can actually see not only what the DNA is within that particular cell, but also what the expression pattern is of the genes that are expressed within that particular cell. And I think that combination of genetic information about epigenetic state and gene expression combined together potentially in a single cell setting is what will be required to tease apart some of the real complexities of diseases that affect not just one tissue within the body, but one particular cell type or a subset of cells within the body.
Thank you very much, and thank you, Lynn.
Thanks, guys.